Bori as the chairman of the session. So we start now the last of the 3 days of the meeting, and I would like to ask Gary Felsenfeld to give his presentation.
Thank you very much. I'm really delighted to have been asked to join this meeting and see many, many old friends and colleagues again. So I'm going to talk about chromatin structure, really, which is what interests us, and I'm going to try to relate it to the issues of this meeting. And of course, the connection is through epigenetics. And I'll remind you that in 1930, Hermann Muller described what he called ever-sporting mutations in Drosophila, which involved, as he realized, chromosome translocations or displacements, as he called them. And he noticed that the genetic material, whatever it was abnormally arranged, but otherwise the chromosome patterns were similar, and of course there was a phenotypic result. This is the first example of what we now call position effect variegation. He realized that the genes were intact, but arrangement mattered. So it's obvious to everyone that now stem cells will contain the same DNA as the cells that result, although the proof of that is actually a work of Gurdon's, Lasky and Gurdon, not so very long ago, to show that the DNA complement in the somatic cells is essentially all the same. But at the beginning, let me say, and you have to keep in mind for the rest of the talk, and maybe even after, that although different complement of genes may be expressed in each cell type, the selection of which genes are turned on and off ultimately has to depend on the binding of sequence-specific transcription factors. This should be obvious. That is to say, there has to be a way ultimately to decide how to turn the genes on and off. That has to involve mechanisms, mainly proteins, that can recognize regulatory elements near the genes and select them. But, so that's necessary, but in the higher organisms, it's not sufficient. And it's the lack of being sufficient that we're really going to talk about today. So, the formal definition of epigenetics is that it's concerned with the transmission through cell division, or maybe in some cases through the germline, of phenotypes that are not determined directly by DNA sequence. So the modern ideas about this phenomenon probably started with Riggs and Holliday about 35 years ago, who proposed first that DNA methylation could serve as an epigenetic mark, and then in more recent years, as we'll see, then many investigators have suggested that histone modifications can also be epigenetic marks, although that's, I would say, we'll go through this, still needs to be demonstrated. Okay. So what are the mechanisms we know about? I said DNA methylation, possibly chromatin structure through histone modifications, and formally speaking, prions also would fit this definition. At least people speaking about prions always show up at meetings on epigenetics, so it's an so far as I'm concerned, that's as much an operational definition as anything. So all of these can persist through cell division, and as I say, some evidence for persistence through the germline. And here's a caution, another caution. Although epigenetic states are maintained by epigenetic marks, these marks have other uses, so they may just be part of a particular biochemical apparatus, and but one tends to refer to them all as epigenetic marks. It's just too much work to keep reminding people that not always do these, not in every situation do these have the possibility of being transmitted. In any case, here's the simple propagation mechanism just to remind you, that goes through DNA methylation. You start with, typically in vertebrates, the preferred site for methylation is a C next to a G, and both strands have CpG and they're both methylated. If you replicate, the new strand doesn't have the methyl group, but an enzyme called DNNT1 will actually recognize this configuration through other proteins and then put a methyl group on. So now you've copied into the next generation. And that clearly does happen. Not too much argument. But now we turn to the histones and histone modifications, which is really most of what I'm going to talk about. So this is the fundamental subunit of chromatin. It's the nucleosome. This is a structure from Tim Richmond's lab some years ago. And it shows 2 superhelical turns of DNA, about 165 base pairs, wrapped about an octamer of positively charged proteins called histones. It's an octamer with 2 each of 4 kinds of histones. So 2A, 2B, 3, and 4. 3 and 4 form the central core as a tetramer, and then 2 dimers of 2A and 2B on the outside lock the second superhelical turn in place, mostly inside, as you see, but very importantly, largely the N-terminal tails are protruding from the structure and available in the environment. Here's a space filler, and you can see, for example, some of these tails actually are protruding through the DNA gyres. And they're outside. Okay. And they are subject to chemical modification, and that's how they play their role. So the lysines in these tails can be acetylated or ubiquitinated, sumolated, methylated. Arginines can be methylated and ADP ribosylated, among other things. Of course, serines and threonines can be phosphorylated. And this is a summary as of 2007 of the known modifications of the various, that's H3, for example, that's histone H4, and you can see, you can get all kinds of modifications, and they occur in vivo in all sorts of permutations and combinations, and those have been referred to as a genetic code, but that's a bit more complicated than that. Codes usually have a one-to-one correspondence, so you can decode them. But these things, the same modifications can have different significance under different situations, biochemical situations or particular genes. And so you can't simply read it off and say, this is an active gene and this is not. Various combinations are maybe specialized for particular situations. But there are some generalizations. Mostly, and for example, monomethylation of lysine 4 on histone H3 is associated with enhancers. And in fact, it's routine now to look at genome-wide data and ask for this signal, and that's likely to be an enhancer. Likely. I think it would be a big mistake to just be certain about that. Lysine 4 trimethylation, same residue on histone H3, is associated with promoters. Lysine 36 trimethylation, the transcribed regions, the coding regions. Very importantly, histone acetylation at multiple sites in 3 and 4 is almost always associated with gene activation and open chromatin. On the other hand, these H3 modifications, lysine 27 trimethyl, lysine 9 trimethyl, tend to be associated with silent or heterochromatin. So those signatures are useful for those of us who study these things. But what I want to talk about now is, since we're talking about possible epigenetic phenomena, is how you can propagate a chromatin state or modification along a chromatin strand, first of all. So it was proposed some years ago, it was observed some years ago, that you could propagate lysine 9 methylation, which is a silencing modification, along a chromatin strand because if you have this site methylated, it will bind a protein called heterochromatin protein 1, HP1, and that in turn recruits the enzyme that makes this modification. So now if you bind here, you can methylate the next one, and then you can methylate the next one, and so you can propagate. And that will actually continue, or could continue, unless you meet a boundary element. We'll talk about those in a while, that could stop this processive thing. There has also been proposed that you could propagate active chromatin, because it is known that acetylases are actually preferentially bound to acetylated histone tails, and so the same kind of mechanism here. So it's easy to extend that kind of model to the replication fork, and that's been done. It's proposed at least. And the idea here is that after replication, it's known that you randomly deposit new nucleosomes on both sides of the fork, interspersed with the old ones. So if the old one is methylated here, then the same mechanism. Here's HP1, there's the enzyme, and it will put a methyl group on the adjacent nucleosome. So now you end up with both strands still carrying the silencing marks. A lot of interest has focused on the so-called polycomb group genes, first identified in Drosophila. And in Drosophila, they were first shown to maintain repressed expression patterns of many developmental regulatory genes that are turned off first by specific factors earlier in development, and then these are kind of placeholders that come in and shut the whole thing down by heterochromatization. And these function by recruiting cofactors that modify the histone side chains in the N-terminal tails, and in particular lysine 27, we now know, trimethylation on histone H3. Okay. And I should just mention that polycomb is a lot more versatile than that, we now know, but this is still an important function, obviously. This is from a recent paper from Danny Reinberg's lab that shows one of the two polycomb group complexes, PRC2, is, as usual, a multi-component complex. And here's the enzyme that lays down the trimethyl mark on lysine 27, the so-called set domain protein, EZH2. And what this group discovered is that this part of the complex actually can recognize the trimethyl mark, and when it binds, it actually changes the set domain conformation in such a way as to activate the trimethylation activity. And so, just as in the slide I showed you before, you can, if that complex binds after replication to the K27 trimethyl mark, it's likely to activate the set domain, and the next one will get trimethylated, and now you copy the silencing mark. Okay. Quite recently, in fact, this came out just a week ago, I think, or two, Valerio Olando's group has looked at what happens in flies just prior to cell division, and what they find is that the PRC2, the polycomb group complexes, rise in abundance dramatically just in early S phase, or just before S phase, and the suggestion is that essentially reinforces the K27 marks and then sets things up for that kind of that copying pattern copying mechanism I showed you on the preceding slide. So there's a lot of work still to be done, but that's the latest position. So now let me talk for a bit about histone variants. So this is the histone octamer, and there's the so-called tetramer, somewhat more stable, quite a bit more stable, and then the histone 2A, 2B dimers, which rather exchange freely. We don't deal really with only one kind of histone, H3, There are variants of histone H3, there are variants of H2A, they differ. The histone 3 variant, H3.3, actually only involves 4 amino acid differences, but that makes quite a lot of difference in its properties. And this histone tends to be associated with transcriptionally active domains. H2AZ tends to be localized at promoters and elsewhere. H3 containing nucleosomes are associated with transcribed genes. 4 years ago or so, Chinwen Jin in the lab identified this hierarchy of stabilities. So nucleosomes containing this pair of variants tend to be least stable. They can be disassembled at moderate salt concentrations. The 2A, 2B come off. These are somewhat more stable, but still relatively unstable compared to the garden variety nucleosome. And now we're going to see what the consequences of that possibly may be. And this is work from John Gurdon's lab, in which he's going to demonstrate epigenetic memory. So he is going to have a donor embryo, these are xenopus, and he is going to take endodermal cells. You can take the nuclei from those and do a nuclear transfer into an enucleated egg. And now he's going to make a new embryo, and the animal region should not express this gene. But in fact, what he finds that is that it does. So that's a mistake, an epigenetic mistake, in which this region misexpresses these cells. And if he then takes again those nuclei and transfers them again, he gets the same result. And what he shows is that this is dependent upon H3.3 levels. So if he knocks down H3.3 levels, then this can be suppressed. You don't get this phenomenon. If you overexpress H3.3, there are more epigenetic mistakes made. So he thinks that H3.3 is an important determinant, epigenetic determinant, and other people have proposed this kind of model. So it's been proposed that the H3k4 tetramer can actually come apart during replication into dimers, and here they are. And then what you need to know is you never see this kind of mixed tetramer. So if you have dimers here that are H3 containing, then when they finish assembling, you'll have the same unstable nucleosomes on either side of the replication fork, and that will tend to maintain the activity of that gene through the next generation. This is John Gurdon's version of that, in which the H3 recruits histone modifying enzymes that do the same thing. Or that may be a more detailed mechanism, but the idea is the same. So now let me talk about some work of ours recently on condensed chromatin domains and how they're established and maintained, work of Keith Giles and Rodolfo Giolando. And this starts with a favorite locus that we've used for many years, which is the chicken beta globin locus. We don't need to worry about what's down here, but in this region was a long stretch of heterochromatin. And we've been studying this for a number of years for a lot of reasons. But for these purposes, the important thing is we knew that all the CPGs in this region, 16 kb long, the DNA is methylated. Outside, it's not. And there are lots of sites for the restriction enzyme HEPA2, CCGG is the motif, but it won't cut if this is methylated. So we treated the nuclei with HEPA2, and that floats out this fragment. And now we can do physical chemistry, we can run sucrose gradients, and PCR, we can study all its properties, but I'm not going to talk about that. I'm going to talk about the control. So the control was to repeat the experiment with a different restriction enzyme, which also cuts CCGG, but it doesn't care whether this is methylated or not. And what we expected is we get tiny fragments. So we did the experiment, and much to our surprise, we got not so much cutting. This is really still quite resistant. So here's the sucrose gradient sedimentation. That's the intact chromatin, quite a lot of it intact, and quite a lot of it may be cut once. And the reason is this is such a compact structure that the restriction enzyme just can't get inside. So how does this happen? And I'll take you quickly through the experiments that we have done. And then I'll explain why they're interesting. So there's a low but measurable level of transcription across this region. Very low, but we can see it. And we wanted to know whether we could disrupt this structure by altering the histone modifications. So we used an enzyme, a compound called trichostatin A, or TSA, which inhibits the histone deacetylase. And so since these marks are turning over all the time, if you inhibit the deacetylase, you get a rise in histone acetylation levels across the region. Not only that, but you see big, almost 7-fold increase in one case across the region in transcripts. So the thing appears to be opening up, and we can confirm that because if we digest with MSP1, that dark blue is what I showed you before. Now everything is moved down to smaller fragments. So now the chromatin's opening up and you can digest it. So now here's the thing that you need to know in order to see why this is of interest. A lot of work on heterochromatin formation and maintenance has been done in fission yeast S. pombe, and a number of labs have done great work, but Shiv Grual and the National Cancer Institute in particular has worked this out in great detail. And this is not the most up-to-date version, but it certainly has the essentials. So this is a centromeric region, say. So it's constitutive heterochromatin. It gets transcribed at low levels during S phase. When the chromatin's opened up, you transcribe it, and you make double-stranded RNA, and an enzyme called dicer cuts that into small pieces, about 25 base pairs long. And then one of the two strands gets stripped off and fed to this protein called argonaute, which is part of a larger complex. And that RNA then targets it back to this DNA. And then this, in turn, recruits all the histone modifying enzymes. That's the yeast version of HP1, for example. You get all the negative modifications and restore the silencing. And really the questions we've been asking in the piece of chromatin I showed you is from chicken, is whether chickens and yeast have any similarity. So I've set this up by showing you what happens if you fiddle with the histone modifications. So now let's see whether the same mechanism that works in yeast works in chicken cells. So we knocked down with RNAi, we knocked down dicer, there is one in chicken, and indeed histone acetylation goes up, and all the other marks that are characteristic of active chromatin go up, and the ones that are characteristic of silent chromatin go down. Transcript levels go up almost an order of magnitude. And if we actually do a chromatin immunoprecipitation, we find that argonaute is bound across this region, but it's dicer dependent. If you knock down dicer, then you lose argonaute binding. And most interesting, if we now do the MSP1 digestion, what we find, the best summarized over here, is that we also open it up to MSP1. So in the control, about 35% of the chromatin is intact, but if we knock down argonaute or dicer, that drops in half. So again, that opens the chromatin. So our picture is that our chicken heterochromatic domain is open for transcription during S phase. The dicer cuts the RNA that's made into small pieces. We've seen them. That gets fed to argonaute, which binds the RNA and targets it back to the chromatin, and we end up in the next generation with silent chromatin. So this is in principle a way you've propagated a chromatin structure, and the question of whether you can influence this in a certain way to disrupt it, and it won't regenerate remains to be seen, but it's certainly a possibility. So the last part of my talk is going to be about something else. It's going to be about long-range interactions and large-scale organization of the nucleus, which is what we're really most interested in now. So specific contacts between distant sites produce a defined architecture, and specific DNA binding proteins help stabilize such interactions, and in particular, the protein CTCF plays a major role in this organization. Dr. Ota mentioned CTCF on the first day of the meeting. This is an 11 zinc finger protein, and it's ubiquitously expressed and had been thought to be somehow involved both in activation and silencing of gene expression, but it's not a transcription factor really. It has a different function. It functions as an insulator, and the kind of insulator it is described here. Suppose you have a green system of promoter and enhancer and the purple system, and you want this, of course, to activate that, but you don't want the green one to activate the purple one. If you put a CTCF binding site in between, then this enhancer will no longer interact with this promoter, but it will still work on that one. And in fact, that's what we found. The most dramatic demonstration of the properties of this kind of system we then identified in this well-known imprinted gene system. This is the Igf2 H19 system, insulin-like growth factor 2. It's imprinted the maternally transmitted allele. Igf2 is off, H19 is on. In the paternal allele, it's the opposite, Igf2 on, H19 off. On the paternal allele, this imprinted control region, the CPGs, are methylated, but not on the maternal. And what we showed was that in mouse there are 4 CTCF sites, and in humans there are 7. And the effect is that this enhancer, which is required to activate IGF2, can't do it. It's blocked. But when you methylate, you lose CTCF binding, and now you have the regular activation. So that's the principal mechanism. There are subsidiary mechanisms, but this is the main one for imprinting at this locus. And Shirley, we and the Tillman lab published this side by side, and then Rolf Olsen's lab in Uppsala separately. But okay, so the question is, how could this work? Sorry, I have to go back. Here we go. So we suggested, what we observed actually is that CTCF has lots of interactions, we know, but one that was of interest was with a protein called nucleophosphine on the nucleolar surface. We suggested that if this interaction tethered the CTCF, you'd create separate domains, loop domains, and if the enhancer was on one and the promoter on another, then you'd get this kind of blocking effect because they wouldn't be able to touch. Alternatively, you just can imagine CTCFs touching each other directly. The topology is the same. And in fact, that turns out to be pretty well the way it works. But we now know more. It can work. CTCF can mediate either enhancer blocking, and there is the enhancer outside the loop, or it can bring an enhancer and promoter together, in which case you actually activate. Depends on the local conformation. The main thing is CTCF stabilizes long-range interactions. The method for measuring those interactions, I'll just briefly see the very powerful method invented by Job Dekker some years ago. You formaldehyde cross-link nuclei, you then restrict, you get rid of that big loop, and then you religate. And now you can actually see whether these two distant sites are in contact, and you use PCR, or if you're doing a more general survey, you'll use genome-wide sequencing. And we now know, for example, that there are long-range contacts all through the nucleus that are very important for function. Here's a brand new paper. This is a Hox locus, and you can see that this domain, there's self-interaction. This domain, there's self-interaction. There's no cross-talk to speak of, and there's a CTCF binding site right in here. Okay. The last couple of slides is just to show you what kinds of things we are now thinking about. This is the human insulin locus, and we're interested in what long-range interactions could have regulatory implications. This is the work of Hsiung, and we're working with human islets. Here's Igf2 H19 I talked about. Insulin is just upstream. And we asked what kinds of interactions occur between insulin and other sites on chromosome 11 in humans. This is the result, and we can see many interactions. There's insulin promoter, and here's a gene called SYT8. And both of these have CTCF sites, I should say. And I'm just, in this last slide, going to summarize a lot of work. What we found was that, in fact, over about 300 kilobases separation, CTCF and SYT8 make physical contact, and that is dependent on the addition of glucose. And when the contacts go up, SYT8 expression goes up. If you target the insulin promoter so that insulin expression goes down, SYT8 expression goes down. If you take away CTCF, again, SYT8 expression goes down, but insulin is unchanged. And SYT8 itself, what it did was not known, but we found it actually affects insulin secretion. So what happens is you have a physical regulatory network over long distances in which the physical contact, it's a kind of transvection, the physical contact will actually, between insulin and its target gene, will actually serve to stimulate the target gene, which in turn will stimulate secretion of insulin. So it's a physical regulatory mechanism. And the important lesson is that physical networks within the nucleus can correspond to new kinds of regulatory networks. And the epigenetic questions in which we're now very interested are long-range interactions preserved during replication and mitosis? And does CTCF serve to establish a scaffold that survives cell division and is important to establishing the specific nature of the cell in which at which you're looking, because these patterns are different in different cell types. So at that point, I'll just say that the work I've described rather quickly was done by Hsiung on the one hand and Rodolfo Giolando on the other, and Keith Giles now at the University of Alabama. So thank you very much.
We are just a little bit over time, so I think, well, the points raised are so important that, well, we are going to discuss them during the discussion at the end of the session. So the next speaker is Dan Hartl, and he's coming to the podium.
First, I want to thank Giorgio Bernardi for his efforts in organizing this meeting and for inviting me to give this talk. Secondly, I want to thank you for your attendance. I don't want to start by insulting every male in this audience, but I almost have to. And the reason is that we share a Y chromosome. And the Y chromosome is widely regarded as degenerate, gene poor, junk rich, transposon riddled, linkage-locked, a rotting genetic wasteland with no SNP variation. That's us, right? Okay, I mean, and this, and the only role for this chromosome, it's widely assumed, is to serve as a pairing partner for the X. And yes, it carries genes in mammals for sex determination, and in Drosophila it has a handful of genes for male fertility. But other than that, the chromosome is assumed to do nothing important. And that's why I say I'm sorry that we share this chromosome. And I'm going to suggest that view of the Y chromosome is wrong, that it's doing a lot of things we didn't know about, and we still don't know how exactly it does it. But the chromosome I'm going to talk about is not the human Y, but the Y chromosome of Drosophila. And here's what it looks like. It's 41 megabases of DNA. Now, for a chromosome, that's nothing but junk. That's an awful lot of DNA to carry around. And you'll see it's almost as big as the X chromosome. Here's the X. Here's the Y. The purple is heterochromatin, which Gary talked about. The Y is exclusively heterochromatic, but the X is about half euchromatic and the rest heterochromatic. The Y chromosome, as far as we know, contains the following material. There are a number of fertility factors, these K factors across here, and very large amounts of microsatellite DNA of various kinds of sequences. There are ribosomal DNA, cistrons, at least in Drosophila melanogaster, but not even in closely related species. I'll talk about these a little more later. Some other repetitive elements here of various kinds. These little tiny triangles are the parts of the Y chromosome that we know the sequence of. And the rest of it is complete terra incognita because the material is either not extractable, not easily sequenced, but certainly not assemblable. So it's an interesting chromosome in which the most modern sequencing technology still cannot reach it. What I'm going to tell you, I'm going to give you some evidence, is that Y chromosomes in natural populations vary from one Y chromosome lineage to the next. And these Y chromosome lineages differ in their effects on the level of expression of hundreds of autosomal genes and X-linked genes. That different Y lineages differ in their effects on these genes, and that closely related species show divergence in the Y chromosome. I'm getting feedback. Are you getting feedback? Okay, so is this any better? Yes. All right, thank you. So here's the experiment that set this off by Bernardo Lemos and Luciana Araripe. The easiest thing in the world for Drosophila geneticists to do is to make chromosomes that are to make strains that are identical for everything but the Y chromosome, because all you have to do is backcross for 2 generations to marked females, and because there's no recombination in Drosophila males, the chromosomes are passed on intact. And so Bernardo and Luciana took a set of Y chromosomes, some from temperate climates and some from tropical climates, and one from an old laboratory strain, and simply introduced these Y chromosomes into a standard multiply marked genetic background, and then did old-fashioned microarrays. Now I say now old-fashioned. Nowadays you would do this with RNA-seq. Old-fashioned microarrays, and they discovered these 1,000 or so X-linked and autosomal genes that show significant differential expression according to the origin of the Y chromosome. Now, if that were just junk, some random differences among the Y chromosomes, you you wouldn't necessarily expect any particular classes of genes or patterns of genes to be affected. But in fact, you do see some patterns. First of all, the genes that are affected tend to be more expressed in males than they are in females. That's what you'd expect, because the Y chromosome is present only in males. The genes that are affected are, the genes that are primarily expressed in females are much less affected by the Y-linked variation. Genes that are more divergent between species are more likely to be affected by differential Y chromosomes. And genes that are polymorphic in expression level within a species are more likely to be affected by the Y chromosome. The question that arises then is whether there are any phenotypic effects associated with this genetic variation due to the Y chromosome, and I'm going to give you several examples of phenotypic effects that are. This is a study of Jean David's performed some time ago, in which they studied the thermal tolerance of spermatogenesis. Drosophila is exquisitely sensitive in its spermatogenesis to temperature. A change of only a few tenths of a degree centigrade will drive male sterility. And Jean's group studied a chromosome from India and one from France, a tropical versus temperate climate. And you'll see that the thermal sterility of the Y chromosome in the French background is quite different from the French chromosome introduced into the other background. And when he did successive back crosses to the other strain, he discovered a big change in temperature back toward the original thermal stability. This goes toward India, this goes toward France, and then it levels off. But about 50% of the total difference in thermal tolerance of spermatogenesis is due exclusively to the Y chromosome. It's not autosomal in effect. Pan Jiang in my lab studied that effect, and she discovered that there were genes at the base of the X chromosome near the heterochromatin that were responding to the Y chromosome, French versus India, that also accounted for a large part of this variation. So you can affiliate particular regions of the genome with thermal tolerance of spermatogenesis. There's another phenotype that's associated with the Y chromosome, and that came out in this experiment as well, and that's a phenomenon called position effect variegation, in which if you take a chromosome that normally lives in euchromatin and make a chromosomal rearrangement where you pop the gene into heterochromatin, say by an inversion, then what happens is in cell lineages, the heterochromatin tends to spread in some lineages farther into the euchromatin than others. So the genes that have been transposed to heterochromatin show variegated expression. And you find in the case of the white eye gene, some lineages of the variegated position effect have a lot of white gene expression, giving you these largely red or orange colored eyes. And you get other lineages that have largely white eyes with just a few spots of red here and there. And what Panban found was that in otherwise completely isogenic strains, the Y chromosome from the Indian strain allowed white gene expression to a much, much larger extent than the Y chromosome from the French background. The Y chromosome also interacts with autosomes and X chromosomes, as demonstrated by these experiments from Chippendale and Rice, where they studied 20 Y chromosomes in 3 different genetic backgrounds and they found huge differences in male reproductive success among these chromosomes. The Y chromosomes had no additive effects of their own, as you'd expect, but there was significant interaction across the genetic backgrounds and individually within the genetic backgrounds. You can see this in another way, where if we just rank the reproductive success of the males, these lines now connect genetic backgrounds that have the same Y chromosome. And you see this Y chromosome does terrifically well in this genetic background, but is terrible in that genetic background. This Y is terrible in that genetic background, does very well in that genetic background. So there's all of these quite unpredictable effects of the Y chromosome interacting with the genetic background on male fertility. We wanted to see whether this effect extended to closely related species. These are now experiments of Tim Sackton, in which he created flies that had the entire genetic background of Drosophila simulans, and either had the Y chromosome of simulans or the Y chromosome of Drosophila seychellia, a very closely related species. And then he did studies of gene expression in these lines, and he did studies of male fertility. And here are the studies of male fertility. These animals are given a fresh batch of virgin females every 2 or 3 days. And so they're inseminating as fast as they can. They live what's called a short but happy life. This guy died right there. They all look pretty ragged at the end of this experiment. So here's the story. The fertility in the Y chromosome introgression drops about 40%. There's a huge effect on male fecundity. There's also in Drosophila 2 kinds of sperm competition. One is whether if a male mates with a female first and then another male comes along and mates, whether the second male can displace the sperm from the first male. So you can have the introgressed male mating first or the introgressed male mating second, and ask whether once the sperm are in there, they stay in there and tend not to be displaced, and whether when the introgressed male is the second male, it's as good at displacing sperm that are already there as the control male. And in both first matings and second matings, the sperm competitive ability either to stay in the female or to displace sperm that are existing in the female, the sperm competitive ability of the introgressed strain is significantly smaller than the competitive ability of the control. So there is clear evidence of very different male reproductive performance depending solely on the origin of the Y chromosome. What's going on in regard to gene expression? Interestingly enough, there are 55 genes that are significantly downregulated in the introgressed lines. Here's a measure of specificity of expression of those genes in the testis. And basically, it's from a sort of mod ENCODE project where you ask, what are the tissues where these genes are being expressed? What proportion of these are found only in testis libraries? And you'll see that almost all of the downregulated genes are highly testis-specific in their expression, and almost all of the upregulated genes have almost no testis specificity. So basically what's going on in these introgressed males is the Y chromosome from Drosophila seychellia is unable to turn on the testis-specific genes of Drosophila simulans to the extent that they ought to be turned on. So here's a brief summary of the main observations. This Y-linked regulatory variation affects lots of genes throughout the genomes, hundreds. Different Y lineages have different effects, and by the way, I didn't make a big deal of it, but these effects are also observed in XXY females, which means that the male fertility factors are not involved in this effect at all, because the male fertility factors are not expressed in XXY females. The divergence between Y chromosomes is that heterologous Y chromosomes tend to downregulate testis-specific genes, and that these have major effects on reproductive fitness. What does all this mean? Well, one possibility is that these regulatory effects are positively selected because of their adaptive value in local populations, and the way the Y chromosome escapes from its linkage-locked, rotting characteristics due to small population size is that the effects are mediated by microsatellites, which increase and decrease in copy number very rapidly through time. So you always have enough mutational variation to select for the adaptive copy number. The other possibility is that all of the variation in the Y that we see is maladaptive, and it results from mutation selection balance, and it's just maintained in populations. And I should point out, these are not mutually exclusive. Some of these effects could be maladaptive and others adaptive, and that still has to be sorted out. One model for what's going on is that here are autosomal and X-linked genes that are binding various chromatin proteins to affect their expression. There's a Y chromosome, and according to the composition of microsatellites and other sequences in the Y titrates some of these chromosomal proteins from these other genes and secondarily affects their expression. Now, this doesn't have to be grabbing across the nucleus for proteins. This could be recruiting those genes into a heterochromatic compartment and affecting their expression, as you see in the case of position effect variegation. What are some of the genes that are affected? Well, to make the story as clean as possible, here's a heat map of the genes affected in Y chromosomes from these different origins, the Congo, Zimbabwe, India, and Ohio, in XXY females. So there's no male fertility factor contribution to this. There are about 101 differentially expressed genes in this heat map. What are they? Well, a large number of chromosomal proteins or transcription factors. Here's a heterochromatic protein 1, GAGA factor. Here's a suppressor of variegation, and various other chromatin proteins. So at some level, chromatin proteins are definitely involved in this effect. I mentioned microsatellites changing in repeat number rapidly over time. We have a set of about 15 lines of Drosophila that derived from a single Y chromosome about 550 generations ago. And so those Y chromosomes can be introgressed into a common genetic background, and you can ask, how are those Ys different from each other after only 500 or so generations? Here is one microsatellite sequence. This is a particular one. These are these are proportional to the number of repeats in these different lines, and you see there are very large differences. This is about a twofold difference in the size of the microsatellite repeat in those different Y chromosomes. You also find large differences in the number of rDNA repeats. Here is the number of rDNA repeats among those Harwich lines, ranging from about 150 to about 500. That's a very large difference across 500 generations. And you'll also see there are very large differences in the degree of position effect variegation. Some of these lines show very red eyes, large amount of white eye expression, of white gene expression, others very little. And you'll also see there's not an obvious correlation between the number of rDNA repeats and and the effect on position effect variegation. However, there is some effect. There is a significant effect of on position effect variegation of the number of uninterrupted rDNA units, but it's just barely significant. So it doesn't account for a lot of the variants, but it accounts for some. I'll underline the word uninterrupted here. The ribosomal DNA cistrons in Drosophila are interrupted by a transposon called R2, which inserts specifically at a specific site in rDNA and inactivates the site, transcriptionally inactivates the site. So across these rDNA arrays, there are a number of these sites that are uninterrupted and a number of sites that are interrupted, and those can be estimated in various ways. And here's another correlation which shows the fraction of the rDNA locus that's uninterrupted by those R2 transposons. And now you see the significance of the correlation is much, much bigger. So this is the fraction of rDNA units that are actually functional across these lines. Again, this does not account for a big proportion of the variance among those for position effect variegation, but it accounts for a significant part of the variation. So I want to thank all of these people for contributing to this work. They've done marvelous work, and it's always my privilege to come and be cheerleaders for the wonderful work that my students and postdocs do. It isn't clear altogether what's going on. You can see this is very early in the investigation, but I can tell you that now we've identified one for certain and two plausible sequence motifs in the upstream regions of all of the genes that are affected by variation in the Y chromosome. And so I hope that'll be a thread in this ball of yarn that we can grab and start pulling on and find out more about what's going on. So I want to thank you for your attention. I appreciate it. And I once again thank you, Giorgio, for the invitation to talk.
We have time for a couple of questions, and then, of course, again, the subject will be discussed in the general discussion. Please.
Dan, you repeatedly referred to the explanation of this phenomenon in terms of epigenetic effects, but you just now said that there's a common sequence motif in front of these genes. And insofar as epigenetic effects usually refers to effects of proteins that don't recognize specific DNA sequences, how do you reconcile those philosophically completely different arguments?
Oh, that's easy. Thank you for your question. It's because I composed the title of this talk and the slides long before we discovered these motifs, which came last week, and I didn't change the title.
In other words, if you had time to make the talk up again, you wouldn't use the word epigenetic.
Well, I'm not sure. Since I don't know what's going on, I think, you know, many of the proteins are in common with many of the proteins that are affected are in common with epigenetic effects in other systems.
You showed us 6 proteins, and you said there are 1,000 affected. So, I mean, if you pull balls out of a pot, you're going to get a few chromatin proteins.
Well, I'd rather talk about data than about words. And if you prefer I not use that word, I'll avoid that word.
It's the concept, not the word.
Yeah, okay. What's your feeling, Gary? You're shaking your head. You agreeing with Eric? All right, better be careful.
Thank you very much. So, well, maybe just one more question.
How did you do these double matings, these second matings? Aren't the female refractive to the second male?
The females are refractive for the second male, but if you just watch them, they eventually will mate with the second male.
Oh, yeah. So you just waited long enough.
You just wait long enough, yeah.
Until the male insisted in long enough.
Yeah.
Thank you very much. So we move to the third speaker of the session, Wen-Hsiung Lee.
First, I would like to thank Georgia for organizing this exciting meeting and for inviting me. This work is almost completed done by my postdoc, Yong Wu, and we are not really experts in epigenetics, but we are evolutionists, try to make sense of some of the data. And since we have experts in the audience, I would appreciate comments and suggestions. The first speaker already gave a very good introduction to chromatin structure and gene regulation. I'll add just a little bit more. This is to tell you how different level of chromatin condensation. And the thing is that we have the pack chromosomes, nucleosomes, and then this is a bit on a string chromatin. And I'll explain a little bit about nucleosome as Dr. Felsenfeld has just explained. You have the H3, H4 form a dimer and a tetramer, and then you have a H2a, H2b form a dimer and then octamer, okay? And then about 147 base pair of DNA wrapped around the Histone Octamer as then to form a nucleosome, okay? And so the point is that for transcription of a gene to occur, it requires that the chromatin region to be accessible to the transcription factor, okay? And so gene regulation by chromatin modification, so histone modification can change histone and DNA interaction, and there are histone modifications that can increase chromatin accessibility and enhance activation, or and in the case of depression, it due to decreased accessibility. And Histone Code, some people don't like this, don't think that they exist, but I'll make a little bit of comment later, okay? So it's a combination of histone modification that provide a code for gene regulation. And you have seen this slide, and here I just want to introduce a nomenclature. Okay, so you have methylation and acetylation. These are two kinds of modification that I'm mainly concerned. And so you have, like say, this notation, H3k4 Trimethylation. So H3 refers to the histone, and K4 means refer to the fourth lysine, and trimethylation means the trimethylation of this lysine, okay? And you have another one is H3k9 acetylation. So you have the residue at lysine residue at the ninth position, okay? And so here are some of the well-known histone marks. One is H3k4 trimethylation, and this taken as active activation mark, usually occur in the promoter region, highly enriched in the transcription factor starting site, okay? And here H3k4 monomethylation, usually associated with active enhancers, okay? And so on, okay? And you see that in some case, methylation, monomethylation can enhance in activation, but dimethylation and trimethylation can enhance repression, okay? And so like this one, H3k4 monomethylation, it's a typical marks in enhancer region. and you see thousands of them in a genome, and they are cell type specific gene related to cell type specific gene expression, and it may be different from it's different from this H3k4 Trimethylation, and it may overlap with other marks and have be active or depressive enhancer. Okay, and this is well known that during the process of development, histone mark may change. So here we consider two marks, and a green one and a red one, and show 3 stages of development. And you can see that green marker has a peak here at this stage, and a little bit shift of a peak, and then disappearance of the peak. Okay, so going through the developmental stage, histone mark can change. Okay, now go into evolution. Okay, and I would like to first summarize some of our observations. So some modifications are evolutionarily better conserved than others, and there's a positive correlation between conservation among tissues and conservation between species. And overlapping of chromatin modifications increases evolutionary conservation. And then I'll talk about a relationship between clustering of transcription factor binding sites and the overlap of chromatin modifications. Okay, and the data we use is a genome-wide histone modification, usually the ChIP-seq data. Okay, and we consider 3 well-known marks, and then we use a terminology that if we see the same site between species, we call that conservation. And currently, most data from mouse and human. Okay, and we consider the same. Let's say if we consider liver, to see if the site is conserved between human and mouse. Okay, and we use stability for the so if a modification is observed in different kind of tissues, we say that's a stability. Okay, and mainly stability data mainly now available in human, and because of this ENCODE project, and there is a start. They have used 9 cell lines, cell types. Okay, and here is a comparison between human and mouse. And Hsiung divided the regions into 4 regions, like say endogenic, promoter, introns, and so on. Okay, and so and you can see that this H3k4 monomethylation has a low degree of conservation, regardless of region. Okay, and this may be reasonable. It's more associated with an enhancer, which is more changeable. And this H3k4 trimethylation is well conserved in the promoter region, but in other regions it may be not well conserved in the liver. Okay, and what was surprising to me is that this H3k27 acetylation, this is actually well conserved, even slightly better conserved than this promoter histone marks. Okay, so we consider their degree of conservation, and we can see that different histone marks have different degree of conservation between human and mouse. Okay, and then we want to relate conservation and stability. Okay, so for stability, as I say, we use different cell types. Okay, and here we have a cell type 8 kind of cell types. Okay, and for this means that this modification is found in 7 or 8 cell types. So we group them together. So we have 4 bins. Okay, and then in this is the cell types, number of cell types, and what we call that stability among cell types. And for this is the evolutionary conservation. And you can clearly see that evolutionary conservation increases with stability for all 3 types of histone marks. And also that this is true for promoter, intergenic, intronic regions. Okay, and this is also true for genomic sites with a near housekeeping gene or tissue-specific genes. So it's quite general. Okay. And then we ask the question, what is the reason behind the positive correlation between stability and conservation of histone modifications? Okay, and we think that clustered transcription factor binding site is the reason for the positive correlation. So we consider, so in this x-axis, this is the number of in vitro validate bindings of transcription factor. We consider unique transcription factors, not just number, but types, okay? And so, for example, this will have a cluster of 17 or 18 different types of transcription factors, okay? And so this is a number of transcription factors are clustered, and this is a conservation, and you can see conservation increases with a number of clustered transcription factor binding site for all 3 types of histone marks. And there's also a study among the individuals and cell types, and you can see that stability also increases with the number of clustered transcription factor binding site. Okay, and so in our view, a transcription factor clustering increase TFBS conservation and then increase histone modification conservation. So this is a transcription factor, and whether it's clustered with other transcription factor 0, 1, 2, and you can see that this increase, the conservation increase. And here we can see that, say, for this particular transcription factor, you see in human, and then you ask whether you also see the same site in mouse, okay? And this is that you don't see in mouse, and this you see in mouse, and you can see that if you see that in mouse, then the conservation is considerably higher, okay? So that's why we form our view, okay? And here I can show you more transcription factor, and you see the same pattern, okay? Non-conserved, conserved, non-conserved, conserved, and so on for all 3 histone marks, okay? And here is a conservation of transcription factor binding is higher when clustered with other transcription factors. This is a logistic regression analysis, and the bar height implies the degree of significance. And you can see that between Drosophila melanogaster and Yakuba, you see that the clustering association with clustering with other transcription factor are significant, except for this one. And you see between human and mouse, and this is among humans. Okay, So we think that if you have a cluster of transcription binding, they may have may be open wide region, so that we can have open region, and that is the most stable. Okay, and how come it's not moving? Okay, and here this is a single site, and you have this modification, and you single regulatory site, and here you have 3 sites clustered together, and if you have mutation, then it's gone, but here if a mutation, then it's still open. Okay, so that you have redundancy and tend to maintain the modification process. Okay, so this is our conclusion. So it seems that the same mechanism works for stability and conservation of histone modification. And we think TFBS clustering enhance stability of histone modification and also evolutionary conservation of histone modification. Okay. And this is because TFBS clustering increase TFBS binding conservation and then histone modification conservation. Okay, so now it comes to the question, which is the cause? Okay, so in the short term, chromatin modification can regulate TF binding events. So chromatin modification conservation may enhance conservation of TF binding. However, in the long term, TF binding conservation would be the ultimate cause of histone modification conservation. Histone modification are conserved to support conservation of TF binding. If TF binding is not there, as I show you, if TF binding is not there, there's no sense to open, to have a chromatin modification conservation. Okay, and we also know that prokaryotes don't have chromatins, but nevertheless, their TF regulatory networks can be inherited to subsequent generations. And it's natural to speculate that histone modification have evolved to accommodate growing complexity of TF regulation. Okay, that's about evolution, and then the histone code. Okay, a histone can undergo multiple modifications, and the histone code might be a combination of histone modifications that can give distinct regulatory signals. And one example is that a combination of H3k4 Trimethylation and H3k27 Trimethylation give bivalent domain. Okay, and here this is a color key for the overlapping pattern similarities. And you can see many histone modifications show similar patterns instead of distinct combinations. And so we have a hypothesis. Overlaps of histone modification can confirm stable chromatin state. Okay, so the experimental data is again ChIP-seq data, and we consider a particular histone mark H3k4 monomethylation conservation between human and mouse, and then this is overlaps with these 3 histone marks. Okay, and here means that you have only this modification alone, and here it means it associated with 3 other modifications. Okay, and you can see that the conservation increases with the number of modifications associated. Okay, and so a general trend is that overlapping of histone modification increases conservation between mouse and human, and for these three, and in different cell types. Okay, and then overlapping histone modification and increased conservation of chromatin accessibility. Accessibility is measured by DNA sensibility, DNA S1 sensibility, assay. Okay, so this is a degree of conservation, and the black bar is a conservation, and this is a non-conservation. Okay, and this is overlapping degree, and this is a accessibility conservation. And you can see that if you have a more overlap, the conservation of accessibility is better. And this is among human individuals, and you see better correlation. Okay, and so our explanation is that multiple histone modifications imply redundant mechanisms for open chromatin accessibility. And we have a positive feedback. Increased accessibility allows factors that mediate histone modification to the genomic region, which in turn would allow further increase in accessibility. Okay, and then we want to see the role of overlapping histone modification in a conservation of gene expression. So multiple histone modification we'll see that increased conservation of open chromatin accessibility. And that's I just showed, okay? Open chromatin accessibility at the regulatory sites, such as promoter or enhancer, these are important determinants. So our hypothesis, histone modification with multiple histone marks tend to be stable, leading to stable gene expression across species. Okay, so here the y-axis is the gene expression divergence between primates, and here again RNA-seq data in liver sample. This is transcriptome data, okay, from higher primates, okay, and then the expression divergence is measured by standard deviation of log RPKM. This is a number of mappable reads in 1 kb region, okay? And then the x-axis is the number of overlapping histone modifications around these histone marks, okay? And this is a region we and then you can see that if you have overlapping histones, so these marks are alone, and his overlap with other histones. And you can see that the expression divergence decreases with the number of overlapping histone marks. Okay, so the conclusion is, in our view, simple view is that histone code is a stability code, and overlapping histone modification can increase the stability of chromatin accessibility, and increased stability of chromatin accessibility can increase the histone chance of histone modification. And genes with overlapping histone modification show stable expression across primates, and this may explain why there are so many histone modification overlaps. And concluding remarks that although this is totally computational, but it requires a lot of literature reading. There are actually a lot of publications on epigenetics, and also a lot of data. And in the past, we don't have many between species data, but now things are changing, and I think that this will give us a good chance to look at evolutionary aspects of histone modifications and epigenetics factors. Okay, thanks for your attention.
Well, we have time for 5 minutes for questions.
Okay, okay.
If the sequence conservation is a real cause for these kind of things, then I think this is genetic,
yes,
not epigenetic.
Huh?
Actually, genetic change is the most important factor.
Yes.
So I don't know whether we should call it epigenetic.
No. We are looking at how epigenetic changes, how they are conserved, or whether they change between species. Okay, and not all changes are caused epigenetic changes. Okay, but some change, in our view, that the most important part is the transcription factor binding sites. Okay, that determines whether histone modification tend to be conserved or not.
Yes.
I get the impression that the term epigenetic is used in several different ways by several different people. And I wish somebody would stand up and say, I am the authority on this, and this is the definition I want everyone to use.
As I say, I'm not the person to explain this, so I use a histone modifications.
The trouble is, if you asked that, 5 people would get up, and each one would say, I am the authority, and each would give you a different definition.
Okay, that's good. That's good. Means you cannot criticize me.
But the problem, the problem. I mean, that's my personal experience.
Yeah.
The problem is because you're also clustering transcription factor binding sites at the same time.
Yeah.
I mean, what's clear is that there's a strong correlation. And from my point of view, if I'm now looking for this kind of site, I would probably search the databases for the correlation of the marks that the overlap of the histone modification marks. That would be a very good way to search
for a regulatory site.
Yes, and then you'd find the regulatory sites
good point.
Nearby.
Yeah.
And they're 2 parts of the same picture.
Yes.
And it's better to think of it that way. It's part of a whole mechanism and involving both transcription and histones modifications, and you can't have the gene expression without those modifications because you can't, the chromatin structure at the promoter will not be accessible, or the enhancer will not be accessible. And so it's just part of a complete mechanism and really sort of hard to dissect, and it's not clear one has to really. Would you agree with that? Or
yeah. But we just want to make a point. The most important point is the transcription factor binding sites, the regulatory sites.
Yeah.
And then the modifications come next.
Yes.
Okay.
Fine.
Yeah.
Okay.
Any more questions?
What you just said is the objection to the term histone code. There's only one code, and that's the DNA sequence code, where the binding sites are, and the rest comes next, as you just said.
Yeah. Okay. Thank you.
Well, time for coffee break. Thank you very much.